I'm going to summarize what we talked about last time, and then we're going to get into these two new topics for today. Previously, we looked at how bacteria control transcription, how they regulate whether or not a gene is transcribed or not. Today, we move into eukaryotic gene expression, how we control gene expression, which is mostly the same way that bacteria do. There are a couple of exceptions. We're going to talk about those and the Gal4 regulation system in yeast specifically. Then we're going to move on to something that I hope hits closer to home, which is lactose intolerance and how transcriptional regulation plays a role in whether or not you can digest dairy products. Well, actually, I hope it doesn't hit close to home, but for many of you, maybe it does. I won't bother asking who's lactose intolerant in here, but it might be interesting to find. To start, though, before we get into Gal4 system and yeast, I want to highlight this, one of your submissions to the Rube Goldberg assignment. So come up with some sort of a transcriptional machine that will take air pollution. We were talking about epigenetics last time we met. We talked about air pollution and asthma. So say air pollution is a small molecule, binds some protein in your nucleus. How does that set off a series of events, like dominoes cascading, such that the promoter of the FOXP, whatever, P3, P4, gene is methylated and is turned off? You develop asthma. I wanted to highlight here, this is not anything that you need to memorize. You probably will learn this in Biology 103 if you wind up taking 103, cell biology. This is a system that cells actually use. This is not something that you invented. This is a system that cells actually use to pass a signal from one cell to another, cell signaling. It's called the Hedgehog pathway. You do not need to remember the details. This is just an example of how it really is the case that cells use Rube Goldberg-like machines to actually turn on and turn off genes. So here's the story, briefly. Here's a cell membrane. Right? There are two proteins that are embedded in the cell membrane. One is this receptor molecule. It's called patched, the purple one or whatever color that is. Shown here is the hedgehog molecule, a small molecule bound to that receptor protein. So this is called hedgehog signaling. The hedgehog protein is a small molecule that one cell sends out into the space in between cells that floats around and then it binds to receptors on other cells. So this is one way that cells communicate with each other. And what happens when the hedgehog molecule on the outside of the cell is bound to its receptor, called patched, is that patched adopts sort of a conformational change. It's got this little hook-like shape. It lets go of a protein it's normally associated with called smoothened. Remember, don't memorize protein names. It's not important. So when hedgehog is bound to patched, patched lets go of smoothened. And what smoothened does is it inhibits two proteins, pKa and slim. What happens when hedgehog's not there, just by looking at the picture? How is this different when one cell is not sending hedgehog to another cell? What does patched do? It binds tightly to smoothened, and it represses it. So here's step one of the Rube Goldberg-like machine. So if one cell wants to communicate to another cell, it sends this protein, hedgehog. Hedgehog binds to patched. Patched releases the protein smoothened. So these two proteins no longer bind to each other. Smoothened goes out and removes the protein activity of two proteins, pKa and slim. What pKa and slim normally do, the next step in the Rube Goldberg machine, in normal situations where there is no hedgehog, on the left side here, pKa and slim take this protein called cubitus interruptus, call it CI, and they stick a phosphate group on it. That's all they do. Once that phosphate group is on CI, it gets cut in half by slim. And then what does that bottom part of the CI protein do? It represses. So what sort of a protein function or what sort of a domain is the bottom half of CI? What does it look like it's doing when it goes into the nucleus? What is this protein physically doing? What's that tube it's sitting on? What kind of a molecule is that? Uh-oh. Yeah, it's DNA. So if the bottom half of the CI protein binds to DNA, what sort of a function does that part of CI have? What sort of a protein domain is that? What's its activity? It's the obvious answer. Don't be shy. It binds DNA. So here's a DNA binding domain. So why would that DNA binding domain going into the nucleus and sitting there on a repressor, how does that repress transcription of, as it says here, hedgehog responsive genes? What might that protein be doing by just going and binding to the DNA? What do we learn about prokaryotes? How some of their repressors prevent transcription? Review. What's the enzyme that's going to cause transcription of the hedgehog responsive genes? What makes messenger RNAs? You could say it louder than that. Yeah, RNA polymerase. So let's say we've got RNA polymerase cruising along here, moving that direction. It gets blocked by this DNA binding domain. So there's no transcription, for example. Okay. So this is kind of Rube Goldberg-like. You get protein binding a cell surface receptor. The cell surface receptor lets go of another protein that goes and represses a couple of proteins that normally put a phosphate group and cut cubitus interruptus in half. Okay. When hedgehog is present, what changes in the bottom half of this figure? Is cubitus interrupted? Does it get a phosphate group or not? Here. It does not. No phosphate. So it's not cleaved. It's not cut in half. So that whole protein enters the nucleus. And the whole CI protein acts as a transcriptional activator, not as a repressor. So the start of the Rube Goldberg machine, somebody tipping over that first domino, is whether or not hedgehog binds to that cell surface receptor patch. And then there's a cascade of downstream events that all winds up leading to whether or not a gene or set of genes is turned on or turned off. So cells use very complicated processes to affect a very simple result. One cell tries to send a signal to another cell to say, turn on these genes. This is one way that that works. And this is one of your made-up mechanisms, which is almost the same. One of you invented this, but it works in essentially the same fashion. There's a protein L, and here's, I assume there was no key submitted with this, so this is me interpreting this figure without a legend. So I'm assuming P is air pollution. So air pollution binds to this protein, which is normally repressing transcription of the CA. Is that the California gene? I don't know. So when you're in the presence of air pollution, L leaves the promoter. So the CA gene is no longer repressed, it's transcribed. It produces the CA protein, which activates now the transcription of CAR1, okay, which is normally not transcribed except for when CA is present and turns on transcription. So the CAR1 protein produces this small molecule C. Here's where we get to methylation of the FOXP promoter. So normally, in this student's view, the methyltransferase that methylates the promoter down here is turned off. But when CAR1 produces this small molecule, that activates the methyltransferase, and the FOXP3 promoter is methylated, and there's no transcription of the FOXP3 gene. Right? This is absolutely outstanding. I loved seeing this, and many of the others too. I only have time to highlight one of these made-up Rube Goldberg-like transcriptional regulation machines. This is exactly the sort of thing that cells actually do to turn on and turn off transcription. Lots of proteins, lots of small molecules, lots of steps, activation, repression, which means it's really kind of difficult to understand how genetics actually works in all of its glorious details. But fantastic work here. So that's all review. So this is what we learned about activation and repression of transcription. Today we get into eukaryotes. So we're going to learn about one new type of DNA element that we haven't talked about yet. And then we're going to talk about environment again. In this case, we're talking about dietary environment, lactose. So step one, I want everybody to know what an enhancer is and to be able to describe what it is and how it works. And then we're going to talk about lactose tolerance. Hopefully there will be no demonstrations. Main difference between prokaryote and eukaryote gene expression is highlighted up here. In general, transcription usually happens. That's sort of the default state for a bacterium. The cell actually has to work hard to turn off transcription. Default state, transcription. Genes are being transcribed unless there are repressors there to stop them being transcribed. The opposite is generally true of eukaryotes. Because we have chromatin, we have our DNA packaged into proteins, and so the ground state, the typical situation in a eukaryote, is not to have transcription unless something actively turns it on. And today we're going to talk about one of those elements that's involved in turning on transcription, the enhancer. Up till this point, we've focused on the promoter of a gene in a eukaryote. Right? That DNA sequence that's -1 to -200 or so base pairs, just upstream of the transcriptional start site. That's this area. That's the promoter of a gene. Today we talk about DNA sequences that are much farther away from the gene. Those are called enhancers. This is another way to say pretty much the exact same thing. We've got the promoter, TADA, where the TADA binding protein binds to initiate transcription. And then nearby, there are some other sites, you don't need to know the names of, that bind general transcription factors, or GTFs. So you might imagine these bubbles are two general transcription factors. General transcription factors are proteins that the cell is usually producing all the time, just like TADA binding protein. So those proteins are almost always around to help turn on transcription, to help bring RNA polymerase to the promoter, and to help produce messenger RNAs. Then, on top of these DNA sequences that are in the promoter or near the promoter, then there are the enhancers. Any questions to this point? So what's an enhancer? How does it differ from promoter proximal elements or the TADA box, for example? So like the TADA box, the GC box, and all these other motifs, enhancers have specific DNA sequences. The only difference between an enhancer and every other DNA sequence we've talked about, proteins binding to this point in time, is that enhancers bind to specific transcription factors, not general transcription factors. So, in other words, transcription factors bind enhancers. We're not talking about general transcription factors like TADA binding protein. Those are proteins that work to start transcription of every single gene in a cell. These are specific transcription factors that turn on only sets of genes. The really tricky thing about enhancers is we have absolutely no idea where they are. It's really hard for a geneticist to figure out where the enhancer sequences are because they're not just upstream of where transcription starts. As this says, they could be really far away on the chromosome, upstream, 5' direction, downstream to the 3' direction, or even in an intron of a gene. So it's been relatively difficult for geneticists to figure out where are these specific DNA sequences, the enhancers, that turn on transcription of specific genes. And part of the reason I'm telling you about lactose intolerance today is we're going to see how lactose intolerance is caused by an enhancer that's in an intron of a gene. I have a specific case study. Questions? We're going to move into something that's going to sound very similar to what we talked about in bacteria in terms of transcriptional regulation. But instead of talking about one type of sugar, now we're going to talk about galactose. So how yeast sense a different sugar in their medium, galactose, and how they process it. So for a yeast to use galactose as a sugar, as a carbon source to metabolize, they need all of these proteins. They need Gal2, Gal1, Gal7, Gal10. They use Gal7 again, and all of this leads to the production of a useful carbon source for yeast to put into glycolysis, not glycosis. Glycosis sounds like some sort of disease. I don't know where this figure came from, but it's wrong. So glycolysis at the end, production of energy. So we're going to use Gal4 as a case for how we, eukaryotes, broadly speaking, us and yeast, you know, best buddies, to do transcriptional regulation. So the main point of this figure is to, again, talk about protein domains. Here's a drawing of the Gal4 protein. Okay. These boxes represent something that binds to enhancers. So in the Gal4 system, the enhancers are called upstream activating sequences, but they're essentially a specific type of an enhancer, specific to Gal4. They're called UAS, upstream activating sequences. So if those UASs are DNA sequences, what are these boxes on the Gal4 protein? What are those boxes, what's their role, what's their function? What does it look like from the figure? Those boxes are going to go bind the UASs. So what's the domain? What's the function of those proteins? Just like the very first question I asked that nobody answered earlier. So if you were paying attention then, you should be able to tell me. Yeah, DNA binding domains. Right. Trying to generate themes here, people. So DNA binding domains, those boxes. So Gal4, the protein, has part of its protein that recognizes the DNA sequence that is the UAS, the enhancer. And notice that all of the Gal genes that need to be turned on to utilize galactose as a carbon source have this DNA sequence or multiple copies of this DNA sequence upstream. So Gal7, Gal10, Gal1, Gal2, they all have the same DNA sequence near them, so that the Gal4 protein will go bind near the transcriptional start sites of all of these genes. So then what's the next question going to be? What does the rest of the Gal4 protein do? If you had to guess, and if you didn't flip ahead to the next slide, if this is a protein that regulates transcription, and it binds near the promoter of a gene, what do you think that other part of the protein would do? Part of the protein brings it to an enhancer. Yeah. So the rest of that protein will probably do something that helps bring in RNA polymerase and stabilize it, recruit it to the promoter and stabilize it there for it to start transcription. So we've got DNA binding domains, and those blobs, the bigger oval ones, are activation domains. And that's exactly what they do. They normally work to bring RNA polymerase to the promoter. The problem is that in normal circumstances for a yeast, this Gal80 promoter is messing up the works, or protein rather. It binds to those activation domains and prevents them from bringing RNA polymerase to the promoter to start transcription. So that's what Gal80 does. Normal situation yeast, in the absence of galactose, doesn't need to produce Gal1 because it has no galactose to metabolize. So Gal80 is there to prevent transcription from being initiated. So time to make another prediction. Go crazy. What's going to happen when galactose shows up in the medium? What will happen to this system, do you think, based on what we know about bacterial transcriptional regulation? So that's one possibility. So you might expect that galactose, that is, the sugar, would bind to Gal80. What would that do, presumably? What would happen to Gal80 when it bound to galactose in this scenario? It would let go of Gal4. So galactose you're proposing would be a small molecule that causes Gal80 to change shape. It dissociates from Gal4, transcription ensues. It turns out that's very close to what happens, but this is more Rube Goldberg than that. It's a little bit more complicated. So instead, there's another protein, Gal3, that's involved. So Gal3 also exists all the time in the cell. It's the sensor of galactose. So it's a protein that recognizes galactose and binds to it. And when those two proteins get together, when galactose is present and it binds to Gal3, those two proteins together, or the protein and the galactose, together interact to knock Gal80 off of Gal4. So it's not direct binding of galactose by Gal80, it's galactose plus Gal4, and those together, galactose and Gal3 work together to remove Gal80 from the activation domains. So it's complicated, but it works. This is how yeast do transcriptional regulation. When they sense galactose, this is how they turn on the Gal genes. So, pardon? Yes, so all of these proteins are available. Is Gal3 in the nucleus or the cytoplasm? That's an excellent question to which I don't know the answer, but I'm going to look it up. So the question was, is Gal3 inside the nucleus or not? It would have to be if it's going to interact with Gal80, which is a protein that's inside the nucleus. So what I don't know is how the galactose gets into the nucleus. Right, you wouldn't expect the sugar to get into the nucleus necessarily. Let me look into that. That's an excellent question. Thank you. This is sort of active questioning. Stump the professor. Other questions? Okay. This is the essence of how enhancers work and why it is that they can be so far away on a chromosome, right? Upstream, downstream, in an intron, not in an intron. These enhancers, the DNA sequences that transcription factors bind to, don't have to be right next to the gene because DNA is flexible. So who knows? This is not a question I expect you to answer. I don't know. There is no limit to how far apart those could be. That could be 1 million base pairs, for example. The UAS, the enhancer sequence, could be millions of base pairs away from the gene it regulates. But the DNA bends, and so that enhancer and the regulatory protein it's bound to can physically interact with the Tada box, for example, of this gene, 1 million base pairs away, because DNA can fold back on itself. And that's why it's so hard to figure out what parts of a chromosome are enhancers. It could be anywhere. The sequences of DNA that proteins bind to turn on transcription. We don't just look right around the promoter in the transcriptional start site. So DNA's flexibility helps regulatory elements for transcription be scattered across the genome, essentially. So then, as was hypothesized earlier, the way the activation domain works is it recruits RNA polymerase to the promoter. It's all about DNA-protein interactions and then protein-protein interaction. So now I want to try to make this relevant, maybe not directly relevant to you, but presumably we all know somebody who's lactose intolerant. And you may be familiar with some sort of jokes about lactose intolerance or something like that. Anybody watch Big Bang Theory? No? It's just me? Okay, never mind. I watch all the science nerdy shows. So all about lactose intolerance for you Leonard fans, right? Here's lactose. And there's galactose, right? That sugar we were just talking about in yeast. Galactose is one of the two sugars that makes up lactose that all mammals make, right? Female mammals produce milk. That's the sugar, lactose, that's in breast milk. We can't digest, thanks Mom, the sugar, the food source that's put into milk all the time throughout our entire lives. Some of us can, some of us can't. We've got lactose tolerance, those of us that can digest this sugar for our entire lives, and we've got people who develop lactose intolerance. Presumably, as babies, we all start being able to produce the enzyme lactase, because all enzymes end with ASE, or almost all enzyme names anyway. So lactase is produced by the LCT gene. So this is why we're going to be talking about transcriptional regulation, because you have to have transcription of the lactase gene to produce the enzyme that helps us digest lactose. And it breaks that bond between the two 6-carbon sugars to produce galactose and glucose, which our guts can easily digest. This is all about transcription of the lactase gene. Turns a sugar that's undigestible into two sugars that we can use for energy. Now, if you're a normal, wild type human, when you're a baby, you produce lots of lactase. Your LCT gene's transcribed, the transcripts are translated, you produce this enzyme. Lactose is broken down into these two sugars that you can digest, and you don't get upset stomach, diarrhea, bloating, cramping, whatever comes with lactose intolerance. But as humans age and become adults, somehow our lactase gene, LCT, gets turned off. We no longer transcribe this gene. Why would we want to do that if we had control over how our genetics worked? What's the point of turning off lactase as an adult?
Because you don't consume lactose. So why?
Because I don't?
No, if you didn't, like, why would you keep doing it?
Okay, so if you didn't, so this kind of gets back to human history. How long has it been since humans have been actively consuming milk? Not breast milk, but into adulthood. Does anybody know how many years it's been since we started consuming camel milk, goat milk, cow's milk, sheep milk? It's on the order of a couple thousand years. So not very long in terms of how long humans have been around on the planet. I mean we inclusively, I'm not saying every one of you. But, right, for a long time, we haven't needed to metabolize lactose as adults. It was only since dairying began that we actually were exposed to the possibility of needing to break down lactose as adults. As a baby, absolutely. But once you're no longer a baby and you stop drinking milk, then what's the point of making lactase? So somehow evolution has allowed our lactase gene to turn off once we're no longer babies.
Dr. Ron?
Please. Well, all of you enjoy this slide.
That is like, looking at it from the genetics and evolution perspective, that people that are lactose tolerant have evolved from the people that are lactose intolerant?
Yes. So main point here is that originally we were all lactose intolerant as adults, or our ancestors were. And at some point mutations arose that let us break down galactose as adults. Spot on. So what happens when you're lactose intolerant and as an adult? Apparently your lactose does not get broken down in your gut and that produces wind in your intestine, which is apparently not a socially acceptable. On the other hand, as Christian points out, there are these weird adults who are sort of infant-like as adults because those rare mutants maintained the ability to break down lactose as adults. So they're baby-like in that their lactose genes, their lactase genes are still being transcribed. So these mutant adults break down lactose, they're lactose tolerant. They can digest dairy as adults. And presumably this arose because of a mutation. So there were some people, few people, maybe one person, maybe a few people in the world, who were in cultures that had begun the practice of dairying, producing milk, cheese, yogurt, labna, whatever. And those adults, the mutants, were able to actually digest those milk sugars. So the question to a geneticist is, how did that happen? What's the mutation and how did it change the way the lactase gene is regulated? So geneticists have dated how long these mutations that allow lactose persistence, the ability to digest lactose as adults, how long those mutations have existed. It's only been a couple thousand years since those mutations actually first struck our DNA. And I want to highlight one here. SNP 13910, don't remember it. SNP, single nucleotide polymorphism, means point mutation. So many people, lots and lots and lots of people, have a C at this spot on their chromosome. It's minus 13, 9, 10. What does that mean plus 1 is? I kind of scribbled over it. Here's plus 1. What gene is plus 1? Transcription of LCT, lactase. So there's a mutation 13,000, almost 14,000 nucleotides away from the lactase gene that controls whether or not you continue to produce lactase as an adult. If you have a C, you don't produce lactase. But if there's a T at that position, you do. So single base pair change that's strongly correlated with whether or not you will continue to produce lactase into adulthood. And that single nucleotide polymorphism is not in the lactase gene, it's actually in a neighboring gene, MCM6. That's the crazy part. Enhancer could be located anywhere, even inside another gene.
What about the N's covering up an A? There.
So you're right.
It's not a single, it's double nucleotide.
Right, so there are many differences at single nucleotide positions between these two chromosomes, one that does produce lactase into adulthood and one that doesn't. But this is, so yes, absolutely correct. This is the most common difference between people that produce lactase in adulthood or don't. But there are other mutations that are associated with that. So presumably it's a cumulative effect. If you have this one plus that one plus maybe numerous other mutations that we haven't even identified yet, you're more likely to produce lactase in adulthood.
Dr. Rob,
Yeah.
how do you know if that's just a correlation or causation?
So in this case, scientists have actually done in vitro or in glass, in test tube, studies with different versions of this enhancer, the one with the C or the one with the T, and they actually show that that one nucleotide difference does actually make an effect on how tightly a transcription factor binds an enhancer. So that one nucleotide difference, the way it works, which you'll see in the next slide or so, is that that DNA sequence where that mutation exists is an enhancer sequence. So here's the lactase, just to put it in perspective. Here's the lactase gene. Here's the MCM6 gene. And that's the position of those SNPs, single nucleotide polymorphisms, including the one we were just talking about, minus 13, 9, 10. So it's pretty far away from the lactase gene. And it's in an intron, those single nucleotide changes. So they're not changing the protein that the MCM6 gene produces. It's a mutation in non-coding DNA. So if you're interested in how this works, this is the article I would suggest you go look at from 2009 Human Genetics paper, where they identify these SNPs and how they work. And that C to T change happens right there. That's that minus 13, 9, 10 position. 13,910 base pairs away from the start of transcription of lactase. That one nucleotide change is in a binding site for this protein called OCT1. That's what that dashed line indicates. It's the DNA sequence that OCT1 protein transcription factor recognizes. So there's a transcription factor that binds there, that OCT1 sequence, the OCT1 protein. That C to T change actually improves how tightly the OCT1 protein binds to the DNA at that enhancer, which means that change from a C to a T increases transcription of whatever gene this enhancer is enhancing transcription of, which in this case is the lactose digestion enzyme lactase. So in this, somebody earlier, way earlier in the term said, hey, Dr. Ross, do you have any examples? I don't remember who it was in class. Do you have any examples of beneficial mutations or mutations that aren't detrimental? Maybe this is one of those, right? A single nucleotide change, its effect is to keep your lactase gene transcribing and producing the lactase protein into adulthood. We could discuss whether or not this is a good thing or a bad thing. I mean, maybe you say it's neutral, it doesn't matter. I would say it's a good thing because it lets me enjoy cheese. But me. So this is exactly like the situation we looked at in Gal4 earlier, if we had changed some names. So in the lactase situation, instead of the gal genes down here, that's going to be, LCT, the lactase gene. That UAS in the Gal4 system, what is it for this lactase story? What was the factor that binds that enhancer? OCT1. So that's an OCT1 site. And it happens to be in an intron of the MCM6 gene. And that makes that protein not Gal4, but what binds the OCT1 binding site? OCT1. Which presumably plays the exact same role as Gal4 in the Gal system. It probably recruits RNA polymerase to the promoter of LCT to turn on transcription of lactase. So the concept is the same, it's just the names and the players change. See, I'd like you to think critically for a minute, if you would, please, about the question in blue. What does this all mean? The couple stories we looked at, especially this one about the OCT1 enhancer, what does it tell us about introns and the so-called junk DNA? We talked weeks ago about how we have like 25,000 genes in our genome. Our genome's billions of base pairs in size. Most of our DNA does not take up the transcriptional units, the sequence that actually codes for the proteins we produce. And that scientists have coined this term called junk DNA that means everything else in the genome. So is that true? What is junk DNA?
Junk is an operative word.
Junk is an operative word. So how do we define what's junk? How do we define what's not junk? I guess maybe is a better question.
Something has a valuable purpose.
Yeah, something that's got a use. So up till this point, we've seen, where am I going with this? Jump in. Up till this point, we've seen promoters, sequences that are really close to the start of transcription. We can easily define their use by looking for common DNA sequences, motifs, like TA, where the TATA binding protein binds. What about the rest of the genome? What about introns? Are introns junk DNA? They get spliced out after transcription, sometimes. What about in this case? Is this intron of MCM6 where this OCT1 binding site is located, would you call that junk DNA? Okay, if not, why not? So the MCM6 gene, one of its introns, has an OCT1 site that these scientists have shown partially controls expression of a different gene. So it's got a function, it's got a job, it's got a role, this intron, or part of this intron. So it's not maybe junk DNA, or at least the whole intron is not junk DNA. Somebody has now ascribed a purpose to it. Extend that to the rest of the genome. How would you know if any part of the genome has a purpose? Has a role it plays somehow in transcriptional regulation, translational regulation, DNA replication? We don't. And that's why most geneticists, you'll ask, will say there is no such thing as junk DNA because we don't know enough about what all of the DNA-protein interactions are to be able to say for certain whether any part of any chromosome has a role or not. Some, it's easy. Genes. Oh, there's a transcript, so that must be an exon, so it's got a role. It encodes protein. But for the other 99% of our genomes, we just don't know yet. So we might not jump to the conclusion that everything is junk just because we don't know if there's a function. We just haven't figured out if there's a function at any particular spot in our genome yet. So speaking of human history, just to wrap up this story, this is from that same journal article. So up on the top graph, we're looking at lactase persistence, or how frequently people from different regions of the world produce lactase into adulthood. Who can digest milk as adults, in other words? Where is it highest? It's the red colors. So we've got Western Africa, we've got the Middle East, and my territory, my homeland, Northern Europe. If you didn't know, that's where I'm from, or my family is from anyway. So these are three areas in the world where lactase persistence is most common. If you compare that to the regions of the world where this single nucleotide polymorphism exists that enhances OCT1 binding, etc., where is it most common? That mutation, not the trait, but the genes. If you were going to bet, if you met somebody and they said they were from a certain part of the world, who are you going to say, oh, you've probably got a T in your MCM6 intron? Okay, I'm not saying that would actually happen, but it looks like Ireland is the place to bet on. Maybe parts of Scandinavia. So this mutation is common to a lot of Northern Europeans, but that doesn't explain what's going on in Western Africa or in the Middle East. So presumably this is where some of those other mutations that are not minus 13, 9, 10, but other mutations nearby might have occurred separately in Africans and in people from the Middle East to help them be lactose persistent. So 13, 9, 10 is mostly common to Caucasians. There are probably different mutations that are common to Africans and Middle Eastern folks who are also lactose persistent. It suggests multiple independent mutations in different parts of the world that all developed dairying at different times and on different animals. So as a Caucasian, as it says here, I guess, I don't know if I told this DNA genotyping company that I was Caucasian or if they guessed from my genotype. Maybe they guessed. But they say I'm a Caucasian anyway. You decide for yourself. I asked this company to genotype me for this SNP we've been talking about, 13, 9, 10, the C to T difference. And let's see, what did they tell me? I have 2 copies of the lactase gene, one from my mother, one from my father. Yes, right. Research has shown that people that have the TT type, like you, so they diagnosed me, they genotyped my DNA. I've got 2 Ts, one that I got from my dad and one from my mom. T was the mutation that allows lactose persistence. So this company tells me that I'm lactose persistent, just based on my genetics. And it's true. I do enjoy ice cream and cheese, and I don't get any of the wind. And so if you're curious about whether or not you think you're going to be lactase persistent or not into adulthood, you could send your DNA off to one of these companies, and they could test you and not tell you, but strongly suggest to you the likelihood that you would be lactase persistent into adulthood. So I want to end by making this a bigger picture story, not just about me and my DNA, because this class is about you and your DNA. These mutations that have popped up around the world in different cultures that allow lactase persistence have only been around for, as was suggested earlier, on the order of about 10,000 years. So not so many generations. And this is all correlation now, but that's just about when anthropologists tell us that people in those cultures started domesticating cattle and camels and began the practices of dairying in Western Africa and in the Middle East. So that's led to a couple of questions and a couple of hypotheses about how our culture and our environment and our genes interact. So here are 2 of the leading hypotheses. You don't need to remember the names of the hypotheses. I'll just put them there for completeness. The first, this one here, we started dairying, and in the presence of a culture where there was milk available, those random individuals that had mutations that allowed them to produce lactase into adulthood were selected for. Now we're talking evolution. So people that were able to access that energy source as adults had better fitness. Maybe they had more kids, so they passed those mutations on to more kids than their neighbors. So that's the cultural historical, that the presence of lactose enabled a benefit when the mutation arose. The other hypothesis is the opposite logic, that the mutations that allowed lactase persistence already existed, and that once dairying began, those individuals were able to take advantage of that by starting to do dairying. So maybe there were little pockets of humans in Western Africa and in the Middle East where those populations already shared that ability to digest lactose into adulthood. And so they started drinking animal milk and started those practices. So maybe something that would be useful to think about, not just in this story, but in general, is which of these bottom points is true? Do our genes drive our cultural practices, or do our cultural practices impact our genes? Hang on just a second. One question? That's correct. So yeah, so different cultures and different populations have different mutations that produce the same effect. Now, for next time, just brief, half a minute. Sorry to keep you late. No reading assignment for next class. It says there is one on the syllabus, but I have abolished it. So we're going to talk about genetically modified organisms next class, and then the last 3 classes you'll see there what we're going to talk about. Okay? So don't do a reading assignment. There is none. It won't be posted on Blackboard. Don't worry about it. We're done with reading for the term.